Dear colleagues, as we unfortunately had to cancel this year's World Congress in Montreal, we thought it would be a good idea to at least share some highlights from the virtual Congress that took place in February. Before the video starts, I would like to announce that the 2022 World Congress in Taipei, Taiwan will take place from June 9th to the 12th. I look forward to seeing you there in person, and now please enjoy the video. Oh, and don't forget to subscribe to our YouTube channel below for free so you can watch more videos in the next weeks. Thank you.
Hi, I'm delighted to speak with you today on our work on the impact of ketamine on intracellular signaling. So to begin with, I do not have any disclosures for this talk. And so while this audience knows a great deal about ketamine, I just want to highlight really the reason why we became interested in ketamine research. So the original work that showed that a single low dose of ketamine, 0.5 mg per kilo infused over 40 minutes, produced a rapid antidepressant response. This was rather unexpected finding, but from our standpoint as a preclinical lab, it really piqued our interest for two reasons. One, the fact that ketamine could produce a rapid antidepressant effect. Up to this point, we thought that in order to trigger an antidepressant effect, it required synaptic rewiring. The fact that ketamine could produce a rapid effect in such a short time frame suggested that you don't have time to redo rewiring, that it's actually possible to trigger a rapid pharmacological antidepressant-induced effect. And second, the fact that ketamine is an NMDA receptor antagonist. Could we actually delineate out how ketamine, specifically through the NMDA receptor, if this was involved, trigger intracellular signaling? Instead of the work that we've done and others had done in terms of trying to understand how typical antidepressants work, where you give them for weeks and you see a change, it's hard to really know how that change occurs. But the fact the rapid nature really suggested we may be able to gain traction to actually put together a framework for what mediates a rapid antidepressant response. So as we were trying to dissect the pathway, what we ended up focusing on was the following pathway. So within your brain, there's two types of neurotransmission, both evoked or spontaneous neurotransmission, sort of the background neurotransmission. So what's shown here is a synapse. With evoked transmission, an action potential comes in, vesicles release glutamate, they bind to the NMDA receptor, calcium comes in, and you activate CAM kinase 2 to trigger a number of downstream effects. But with spontaneous neurotransmission, what happens is there's a different pool of vesicles that release glutamate. They bind to a different population of NMDA receptors. Calcium again comes in, but instead of activating CAM kinase 2, you activate CAM kinase 3, a calcium calmodulin dependent kinase that is interesting because it only has one target. So this kinase can phosphorylate EEF2 and then have an effect on protein translation such that it inhibits it. So if you block these NMDA receptors, calcium doesn't come in, the kinase is not activated, and you desuppress or result in a rapid upregulation of protein synthesis. We actually could show that this was occurring both in vitro and in vivo. So we ask, if this is really true, is this really setting up our signaling cascade? In the sense that is EEF2 kinase, which we postulated was upstream of BDNF, it's driving BDNF expression, and we could show this, but was this really important for antidepressant action? And what we could find is that if you took EEF2 kinase knockout animals, and if we give them ketamine, in wild-type glutamate control animals, you get a rapid increase in BDNF protein within 30 minutes, but not in the knockout. And we characterized these knockouts in a range of behavioral paradigms. And what we found is just deleting EEF2 kinase had no effect on baseline immobility in the forced swim test. But with the controls, again, given ketamine, you get the significant decrease, suggestive of an antidepressant effect that is blocked in the knockouts, showing that EEF2 kinase was required. So similar to BDNF, EEF2 kinase was required, but we went on to show that this was actually driving the BDNF expression. You knock out the kinase, BDNF expression, like we show, is not upregulated, so it is upstream. As we were trying to really understand this, we have like NMDA receptors, this kinase, it's important, it's driving BDNF and other protein synthesis. But what's happening? What's sort of triggering the effect? This is sort of mediating the effect, but it's rather surprising that you can have antidepressant effects out for days or weeks. It suggests a type of plasticity. However, it's well characterized that NMDA receptors, especially if you just look at hippocampal plasticity, you just take a hippocampal slice, you perfuse on an NMDA receptor antagonist, it doesn't elicit any type of plasticity. So how could we explain these long-term effects? So what we did is in terms of the potentiation, we took C57 animals. We were able to induce this plasticity, and then we added NASPAM, which blocks GluA2 lacking AMPA receptors. And you can see you still get this plasticity. We couldn't block it, suggesting GluA2 truly was involved in this potentiation. If, however, we took C57 animals infused on ketamine, we induce this potentiation, and then if we add DNQX, which just blocks all AMPA receptors, we block the effect, showing that this is AMPA dependent, similar to the behavioral effect. We then obtained both GluA1 and GluA2 animals and did work. I'm just going to show you work on GluA2, which was surprising. With GluA2, again, this potentiation, we see it in littermate controls, but not in GluA2 knockouts. And we also could show that GluA2 is required for the behavioral effect. So what we think this means in terms of our conclusion is that we think that ketamine is directly blocking NMDA receptors on excitatory neurons that impact calcium flow, that directly impact the activity of this kinase, thereby desuppressing protein translation, which increases BDNF protein expression, as well as a number of synaptic proteins that is driving the insertion of AMPA receptors. And from that, this novel form of potentiation, which we think is important for the antidepressant effect. And this explains how you could potentially have the longer lasting ketamine effects, because there is a potentiation type effect that is occurring. And it's rather surprising, but the only way to really trigger this sort of potentiation would be through effects on excitatory neurons, at least for what's known. So we think that it really argues for a direct role of ketamine blocking NMDA receptors. And again, it's functional effects on the NMDA receptor that can determine whether you impact this pathway, as I showed you with both ketamine and memantine. So with that, I'm just going to close with an acknowledgement slide of the individuals in my group.
I'm delighted to be here participating in a session on rapid onset and sustained action of ketamine in depressive episodes. My topic for today will be neural networks involved in the action of ketamine. This is my disclosure. I'm a full-time employee of the US government. And this slide summarizes much of the work that will be presented today, but that the theory when we tested ketamine was that NMDA receptor inhibition would bring about rapid antidepressant effects in individuals with treatment-resistant depression. In this study, individuals have failed on average 5 to 6 antidepressant. A single infusion of ketamine in red compared to the control condition in blue significantly improves depressive symptoms within a matter of minutes, lasting a good part of the week. Towards the right, we see response rates to traditional antidepressants, where one takes an antidepressant pill every day for 8 weeks. And in approximately that time, one achieves a 62–65% improvement in individuals who do not have treatment-resistant depression. Very important to mention that the half-life of ketamine is only a few hours, and the dissociative side effect of ketamine is only about 40 minutes. There are several theories on how ketamine might be involved, in terms of its mechanistic properties. One is that ketamine blocks NMDA receptors, which leads to a change in eukaryotic elongation factor. 2, a series of intracellular signaling cascade changes, BDNF translation, protein synthesis, and an increase in spines. Another alternative theory is that ketamine blocks NMDA receptors on GABAergic interneurons. When it does so, it decreases GABAergic inhibition, produces an excitation of pyramidal cells, which results in an evoked release of glutamate, activating AMPA receptors, and also TrkB, intracellular signaling cascade changes, And towards the top right, you see a restoration of spines and synapses within a very short period of time. That's another theory into how ketamine might work. Now, the one limitation of this is, of course, blocking NMD receptors leads to potential of abuse and potentially psychomimetic effects. So can one produce an increased probability of evoked release of glutamate away from the NMD receptor? And the answer appears to be yes. One can do so at mGlu-R receptors with an antagonist, or at the GABAergic neurons with a GABA-A negative allosteric modulator, or with the metabolite of ketamine 2R6R-H&K. All of them seem to produce an increased probability of evoked release of glutamate. In earlier work in mid-2000, in this study, what was shown that ketamine decreases immobility in the forced swim test, but when you pre-treat with an AMP antagonist, MBQX, you effectively abolish the antidepressant-like properties of ketamine in rodents, signifying that ketamine's therapeutic profile seems to be by activating AMPA receptor. All of the mentioned candidate drugs have this property where their therapeutic properties are blocked by an AMPA antagonist. Furthermore, these different compounds increase gamma power, which is in the 30 to 50 hertz range of EEG activity, which is a surrogate measure of this excitation inhibition balance or neuronal synchronization, and potentially could give us a marker of target engagement. Here you see the group differences in ketamine effects on whole brain functional connectivity across 4 striatal seeds. In green is placebo, in orange is ketamine. And up means greater connectivity, down lower connectivity. And you see in treatment-resistant subjects across the 4 seeds an increase in functional connectivity with ketamine compared to the control condition. Whereas in healthy control subjects, you actually see the opposite, a decrease in functional connectivity across the 4 seeds compared to the control condition, which means that there are differences in its response and perhaps at a homeostatic level. Towards the bottom, you see the changes in connectivity correlated with changes in depressive symptoms. And you see at the ventral stratum seed, a very nice correlation between increase in ventral stratum and the dorsolateral prefrontal cortex connection with antidepressant effect, and also improvement in depressive symptoms in other seed regions. Towards here in B and F, you see the relationship between dorsal cortiate and improvement in anhedonic symptoms in a very nice correlation. So here we see both antidepressant and anti-anhedonic effects in the relationship with neurobiological changes at the circuit level. This is to show you that if we induce a stimulus, air puffs in the finger, we produce increases in stimulus-induced gamma power. No difference in responders, non-responders, a significant increase in the responders post-ketamine. Here in a control study, we show the same thing. Responders have an increase in stimulus-induced gamma power, which is related to the peak of antidepressant effects at 24 hours. So to summarize to this point, ketamine does appear to have therapeutic properties, and it peaks within hours. There are metabolites that are actively involved, and some of these are also associated with gamma properties and pose as very interesting candidate drugs for testing in depression. Thank you for your attention.